We now go to irreversible cell injury. Irreversible cell injury, from the word, it's quite obvious, irreversible. The changes that take place in a cell cannot be reversed. It finally results in cell death and the cell getting out of the body. Okay. So cell death is a state of irreversible injury. Now we have different forms of cell death. The cell can die in different ways. It could be autolysis, through necrosis, through apoptosis. This particular sheet is not, this slide is not there. There could be now these two things, no? Gangrene and pathologic calcification. These happen after apoptosis or necrosis. Able to follow me? See, when a cell has to die because of an irreversible change, it could die through autolysis, necrosis or apoptosis. When necrosis or apoptosis happen, that particular tissue could undergo gangrene, a gangrenous change, or it could undergo pathologic calcification. But both of these also result in cell death. So we can say it's a sequence. Apoptosis leading to gangrene or apoptosis leading to pathological calcification. Alright, it's fine. Now, this particular thing is there in your keynote, so don't worry. What is necrosis? Necrosis is focal cell death. Means localized or a particular cell is undergoing death will be called as necrosis. A few terms here. What is gangrene? Now, there is a necrosed area. Over there, bacteria will come. Right? And bacteria causes infection there. The two things combined together becomes gangrene. So what is gangrene? Gangrene is necrosis with superadded putrefaction. These could come as an MCQ. Then there is something called as necrobiosis. Necrobiosis means there is necrosis, necrosis leading to degeneration. That's necrobiosis. Now in necrosis, finally there is going to be cell death. Okay, so in necrosis you are going to have changes in the cytoplasm. And there will be changes in the nucleus. Changes in a way that finally the cell gets disintegrated and dies. Now cytoplasmic changes could include accumulation of your microtubules, microfilaments, or changes in the endoplasmic reticulum and all that. I will be particularly dealing with nuclear changes because there have been MCQs on this and I have seen a doubt on this in Facebook as well. What are the changes that you will see in the nucleus? The nucleus may undergo pycnosis, karyolysis, and finally become karyohexis. Okay? So, pycnosis and karyolysis, there's a confusion, I think. Now, pycnosis is basically where there is a condensation of all the nuclear chromatin or when there is a shrinkage. So, there is an actual reduction or a shrinkage of all the chromatin that is inside the nucleus. That will be pycnosis. Now, if they say fading, if you see the term fading or if you see the term dissolution, then it becomes karyolysis. And finally, if the content of the nucleus is fragmenting and forming clumps, and loosely disintegrating into itself is karyohexis. So are these three terms clear? So you should be looking in for terms like shrinkage or condensation, which will be pycnosis. If there is just fading of the material or dissolution, then it would be karyolysis. Okay? We all know there can be. Now destruction dhara is, what kind of destruction are you talking about? Destruction is a very vague term to talk of. Destruction can be fragmentation, destruction can be condensation. I mean, all these three things are causing destruction of the nucleus. Right? I know, it's a very vague term to use. Destruction of nucleus means all these three are destroying the nucleus finally. But I think he's trying to ask you about the final breakdown of nucleus and dividing. So you can go in for karyohexes because that leads to fragmentation. I mean, maybe the examiner was trying to ask a question where there is an actual breakdown of the nucleus. So I think in that case it could be karyohexes. If that's what is in his mind. I mean, that's what I think of for it. Okay. We know that there can be different types of necrosis. We have 5 types of necrosis, which is coagulation necrosis, liquefactive necrosis, we have fat necrosis and all that. This is also there in your keynotes. Okay. We were talking about coagulation necrosis. The point about this is, now suppose there is a cell which is undergoing necrosis. Yes, there will be death, but the outline of it will be retained. There might be cell changes inside causing death, but the outline is retained. So it says, it converts the cell into their tombstones. Can all of you see the slide? Audio, video, all good for everyone? Okay. Examples of necrosis is what is important for you from an MCQ over here, okay? Where do you see coagulation necrosis? An infarct of the heart, kidney and spleen. Extremely important. Put a star across it. You will have an MCQ over here. So that's where you see coagulation necrosis in case of an infarction in the heart, kidney and spleen. Then we talk about necrosis, also called as colliquative necrosis. Coagulative necrosis. Do not understand? Coagulative necrosis happens whenever there is an ischemia. Whenever there is a loss of oxygen to it, this undergoes coagulation over there and becomes necrosed. But what is the feature of it is that, yes, there is a cell which is undergoing cell death, but the outline is retained. Important is this, infarction. Areas where you see necrosis, for all types of necrosis, it is the examples of it which is an MCQ. For all types, you will find this kind of a chart in almost all your MCQ books, okay? So over there, the MCQ that's frequently asked is the examples of each type of necrosis. Let's go to liquefaction necrosis, also called as colliquative necrosis. Here, there is, the cell becomes liquid type, you know, it's melting down type. That's why it is called as liquefactive. This also happens because of ischemic necrosis, but very important is there is a bacterial infection and it's more commonly seen in the brain. So an infarct of the brain is a very, very typical example of a liquefactive necrosis. Very, very important here. Okay. So an abscess cavity or an infarct of the brain is where there is liquefaction necrosis. Liquefaction, now think of what you see in an abscess cavity. You see liquid, you see pus, you see liquid there. Okay, you may not see that in coagulation necrosis. Okay, next we go to caseation necrosis. You must have heard of this term. It's usually seen with respect to the TB. Tuberculosis, where you frequently see it. Now what happens over here is it's a combination of a coagulative necrosis as well as a liquefactive necrosis. Now what will happen is over here, in case of, if you think of a caseous necrosis, you think of a cell which is undergoing this, you will have bacteria accumulating in the center and it results in a cheese-like formation in the center. That's why caseous means something like a cheese-like material. Okay, that's called as caseous necrosis, very commonly seen with TB, MCQ. And the main reason why this happens, it's all only seen in TB, caseous necrosis. Why does it happen? It is actually because of contribution of the cell membrane of the cell. The capsule of your mycobacterium contains lipopolysaccharide. Its content comes out, okay, and it forms a cheesy material in the center of the cell. So that's caseous necrosis. Then we have fibrinoid necrosis. From the term itself, you can understand it's deposition of fibrin-like material. So you have a lot of fibers being deposited over there. That becomes a fibrinoid necrosis. And where do you see this? Think of most of the autoimmune diseases, joint disorders. You know, you have a lot of fibrin. You have less cells and more of fibrin getting deposited over there. So in autoimmune disorders, in arterioles, in hypertension also you have a wall getting thickened. Thickening of the wall is because of what? At the borders of the blood vessels, you're going to have more of fibrin deposition taking place. So these are examples of a fibrinoid necrosis. Then we have fat necrosis. Fat necrosis, simple. There's going to be fat accumulation. It's a special form of cell death. Where do you see it? Examples of this is acute pancreatic necrosis, traumatic necrosis of the breasts. Breast is nothing but adipose tissue. Okay. So suppose there is necrosis over there, it could be a type of fat necrosis. Are we clear with different types? This chart that is there in your keynotes is very important. You have a lot of MCQs from there, especially the examples. Okay, now let's go to apoptosis. Now from henceforth, the couple of slides that is going to be coming henceforth are not there in your keynotes. And the reason why we have put it now is we have seen a lot of questions coming from apoptosis in the last. Okay, so you can either take a snapshot of it or make sure you can write it down. But I think more than wasting time on writing, please listen to me and understand what it is. You can always watch it from the recording and write it down. And in fact, it is there in all the smaller to the bigger standard textbooks have it. So rather listen to me and understand what it is. Writing it, you can always do it later. Okay? Fine? What do you understand by apoptosis? Apoptosis is nothing but programmed cell death. By heart these three words. Programmed cell death. MCQ here. Now, this apoptosis, it can be physiological or it can be pathological. All of us are clear, right, that during a normal physiologic process also, you need some cells to die and get eliminated from the body. Your epithelium always undergoes a constant cycle, where you have some cells being dying and then even your blood cells for that matter. There is a cycle, constant cycle that is going on. So these different ways of how the cell die, the replication, can involve apoptosis. So let's see what are the places where you see a physiological apoptosis. In case of embryogenesis, some cells die, some cells stay right from implantation of the fetus to birth, right? See, whatever is seen in the placenta eventually dies, right? There is regression of that cells of the placenta after the birth of the child. So even in the beginning, there are some cells that is required only for maintenance of the fetus which eventually die. So during embryogenesis, then during the menstrual cycle, there is an endometrial cell breakdown. Again, this menstrual cycle is a continuous cycle which will require cells to be continuously dying. So this will also be as a result of apoptosis. Then, during a normal immunological reaction, the T lymphocytes are also involved in some amount of cell death. It's not talking about pathological. I told you there are some cells which need to be removed from your body, right? Whenever they found to be either defective or they're aged or whatever. So those is also brought about by the CD8 cells, that is the cytotoxic T lymphocytes. Cytotoxic T lymphocytes is nothing but your CD8, right? The suppressors. So the cell death which is induced because of these is also because of apoptosis. And a routine cycle cell death. So these are where the physiological changes will take place. Now what is pathological apoptosis? Now in cases of your DNA damage or a cell injury in infections or there is an atrophy of the organs, so there's pathology that is involved. In all those cases, whatever cell is dying is happening because of apoptosis. Is it fine? Okay, but this physiological pathological is easy, so I think this should be okay. Let me go to the next slide. We have talked pathways of apoptosis. Like how you see in a coagulation cascade, you have extrinsic mechanism and an intrinsic mechanism. Similarly, in apoptosis, you have two pathways. You can just simply write a note on this. There are two pathways in apoptosis also. We have an intrinsic pathway and we have an extrinsic pathway. Yes, I am frequently clicking so that I can avoid the lag. There seems to be an internet problem today. Can you guys hear me now? Okay. Okay, you can write this down. Intrinsic mechanism and extrinsic mechanism. What is intrinsic mechanism? From the word itself, it happens inside the mitochondria. Yes, Rakini, I understand there's an overlap and a lag, and whenever I do get an indication of it from my side, and whenever that happens, I am clicking on it. But if it gets too disturbing, let me know. I'll have to stop the class in that case. If it's fine and if you guys can manage with it, we can continue. Okay. Yeah. Let me know if it's a real problem, alright? Now talking about the two pathways, we have intrinsic pathway and an extrinsic pathway. An intrinsic pathway is what happens inside the mitochondria. So you can have an MCQ on this in terms of majority of, or where does the process of apoptosis take place. So it takes place inside the mitochondria if it is an intrinsic pathway. Okay. There is also an extrinsic pathway which is called as a death receptor pathway. An extrinsic pathway happens outside the cell. It is basically happening on the cell surface. Whichever, this also again involves your T lymphocytes. So it's an extrinsic thing. Now you can have a look at this diagram that is there. It's describing both the extrinsic pathway and the intrinsic pathway. I will not be getting into the extreme details of the two pathways. Look at the diagram here. All of you can see this diagram. Okay. Let's look into the extrinsic pathway over here. There is something called as the Fas protein. Okay, it's a ligand. It is this ligand which will go and combine on the surface. It again involves the T lymphocytes over here, and the final process that is required to do is activation of caspases. I will discuss the whole thing, don't worry, but just understand the diagram first. There is a ligand. The ligand name is Fas, F-A-S. Okay, it needs to combine with a protein that is inside the cell called as FAD. This requires T lymphocyte. Okay. Finally, it's as long as your Fas is combining with the lymphocyte and it is activating FAD, that's all is what is extrinsic pathway. Okay. Whatever happens after that is happening inside the cell. And the receptor which is present on the surface of the T lymphocyte is called as a death receptor because it's frequently causing your apoptosis. No, it is only involved in causing the apoptosis. We call it as death receptor because it's finally causing death of the cell. Actually, Saina, everybody else seems to be having a slide. Check the internet issue or the flash player from your end. All of you can see the slide, right? Yes. Okay. So like I said, there is only one thing that is required in the extrinsic pathway. It needs a certain ligand to combine with the receptor on the surface of the T lymphocyte. The receptor which is present on the surface, since it's always involved with cell death, it is called as a death receptor. So the FAS will bind with that death receptor, and that's about the extrinsic pathway. Whatever happens after that is happening inside, and the final thing that is required, or the final step that is required to initiate apoptosis is to activate a certain protein called as a caspases 9. This is an MCQ. The most important protein that is involved with apoptosis is caspases. Is this okay? FAD is not the death receptor. FAD is another thing that FAD is not the death receptor. It is inside the cell. Okay? Caspases apoptosis is clear. You will have that as an MCQ. Okay. Now let's look into, yeah, it's too much of a molecular thing, you know, don't get into it. What is FAD and all that. It's going to be too difficult for me to explain. So, I mean, it's not been asked, you know. Caspases has been asked, 2 pathways have been asked. So don't get into too much of detail of it. It will be too time consuming for you. Okay. Next we go to the intrinsic mechanism. Okay, this is what I told you. There's another thing that is required on this is the tumor necrosis factor. The death receptor that we were talking about, no? That on the surface of your lymphocyte you have a death receptor. What is that receptor? It belongs to the family of tumor necrosis factor, TNF. So it's a type 1 TNF receptor. And this ligand is expressed on the T lymphocytes. It's these death receptors which will trigger the apoptosis. Then we go to the intrinsic pathway. Now intrinsic pathway is a little more important because there have been a couple of MCQs on this. I want you all to concentrate. All of you with me, right? Okay. Now first thing, it happens inside the mitochondria. So let's draw one mitochondria there. Now, inside this mitochondria, we have a lot of proteins. Okay? Now, there are proteins inside the mitochondria which will Is there still a voice lag? Okay. Now, there are what I was saying is there are several proteins which are present inside the mitochondria and they fall into separate groups. Okay. Some of them inhibit apoptosis while some of them promote apoptosis. So we have 2 groups. We have promoters, we have inhibitors, and all of these are present inside the mitochondria. Okay. Now, so able to follow what is promoters and what are inhibitors. Hi guys, all of you are back. Is the audio video fine? Can you all see the slide? Let's continue. We were talking about the 2 pathways that is there in cases of apoptosis. So we have an extrinsic pathway and we have an intrinsic pathway. In extrinsic pathway, there you have to remember there is something called as what family of receptor do we have there? What is the name of that receptor and to which family does it belong to? Yeah, the name of the receptor is the death receptor. And it belongs to which family? The tumor necrosis factor, the TNF family. Yeah. Then come to the intrinsic pathway and like discussed, it happens inside the mitochondria. Inside the mitochondria, we have a lot of different proteins and genes that are involved with it. All of these which are mentioned over here are genes which are regulating the apoptosis inside the mitochondria. And all of these can be asked as an MCQ and they have been asked in the past. So this is something you need to make a note of quickly. Okay. These are all regulators, means they regulate. It could either increase or it could decrease apoptosis. So like I said, there will be some genes which will be involved in promoting apoptosis. What are those which promote BAX, BAD, FAS and P53? MCQ here. All of you with me? Able to follow me? Genes which promote apoptosis, BAX, BAD, FAS, P53. Inhibitors means those which will stop or inhibit apoptosis, BCL-XL and BCL2. And just another mention of all the regulators, P53, MYC, RAS, FAS. Okay. No, we've only discussed about the promoters, inhibitors and regulators. Nothing much otherwise, Kamal. Just the names that you see here, no? You need to make a note of these, and these could be an MCQ. What are the promoters of apoptosis? What are the inhibitors of apoptosis? And as mentioned earlier, in terms of the number of proteins involved, the major protein group which is present in the mitochondria is the BCL2 protein. That's a major protein group that is present inside. Now, what I said is, all of these will either initiate apoptosis or they will inhibit apoptosis. Now, suppose it initiates apoptosis. Now, there are several mechanisms that will initiate it, but the final important thing in apoptosis is to activate caspase. In that also, specifically, it needs to activate caspase 9. Which caspase it needs to activate? Caspase 9. MCQ here again. Okay. Let's go back to the diagram. Okay, all of you can see there are two things. The textbook says caspase 9. There is also caspase 3, definitely. I'm not saying no. But it says finally important step is activation of caspase 9. Okay, caspase 9 activates caspase 3. And these bring about a series of reactions which we don't need to know, right? But there is also another important thing that is involved in this which you can also appreciate in the diagram. You see cytochrome C over there? All of you can see that, no? So cytochrome C along with cofactors is also necessary for apoptosis. And all this that I have mentioned to you is MCQ. Are we clear on this? Two pathways, what really happens in these pathways, and what are the promoters, inhibitors, regulators. Isko अच्छे से by heart karo, you will have MCQ on this. Then protein involved, caspase 9, cytochrome C involvement, this has also been asked as MCQ. Can I proceed? Is this clear? Okay. Now we have seen necrosis, we have also seen apoptosis, right? Both of them are causing cell death. But there still seems to be a difference between the two. So differences between necrosis and apoptosis is also an important source of MCQ. You will find that chart in a lot of your books. You can see the difference over here. What induces necrosis? It could be ischemia, oxidative stress, mechanical injury, whatever. The same things can cause apoptosis as well, but in addition to it, it will have to be a stimulus from FAS and TNF alpha. These also involved in the stimulation of apoptosis. Now what happens? The simple difference between the two is, in apoptosis, you're going to have fragmentation. You're going to have that cell breaking down into tidbit. Okay, but that does not happen in necrosis. You know, necrosis is going to have internal cell changes and the cell dying as such without disintegration, you know. You can see the diagram, you can see the changes. You can see how here there is cell lysis, okay. There will be an inflammatory response and there will be engulfment of the cell or whatever. But here you will see that the cell actually breaks down, and then each of these individual fragments will then be phagocytosed. And there will be no inflammatory response in case of apoptosis. But a necrosis will always have an inflammatory response. Okay, MCQ here. You guys able to follow me? Very important is fragmentation, and here no fragmentation. Inflammatory response not induced in apoptosis, it is always induced in necrosis. Another thing, necrosis will have cell swelling. Apoptosis will have cell fragmentation. Okay? Here also you can see these changes. This is again important. You have cell swelling, no condensation of chromatin, and you have a loss of ATP. Okay? It's a passive process. But something like apoptosis requires ATP, becomes an active process. It requires activation of caspases, chromatin condensation, and shell shrinkage. So opposite changes between the two. You can make a nice table of this, and there will be an MCQ from this as well. Is this fine? Can I change the slide? Just take a snapshot of this. It makes it easy.